Hello, and for our final talk this morning, we have Dr. David Lowry, who received his doctorate at Duke University and is now an associate professor of plant biology at the University of Michigan. David's lab focuses on identifying genetic and physiological mechanisms of ecological adaptations and how these contribute to the formation of new species, and then applying these insights into crop plants. They work on several plants, including mimulus and common bean, both impactful JGI genomes. And here he's going to tell us about his work with my favorite North American perennial grass, switchgrass, and its interactions with pathogens. Please put questions in the Q&A box. Welcome, David, and thanks for joining us this morning.
Okay, thanks, Jeremy, for the introduction. I really appreciate that, and I'm glad to be here today and excited to tell you about our work on switchgrass that we've been working on for the past decade or so, but recently really brought in a lot of new work on microbiome, both in terms of pathogens as well as the microbiome that is beneficial to the plants as well. So I'm going to start off by telling you a little bit about the intellectual framework for our research, and then I'll tell you about how we use the setup that we have for switchgrass in order to study microbiome assembly and how the environment and the genetic variation within switchgrass controls that assembly. Okay, so the basis of a lot of the research that goes on in my lab and with people who study local adaptation started out with goat Turrison in 1922, so almost 100 years ago now. And Turrison conducted a series of experiments along environmental gradients in Sweden. And he collected plants from all over Sweden and planted them into common gardens. And from that, he saw that plants from different regions maintained their differences in common garden experiments. And this told him that there was natural selection occurring in those different habitats that drove those differences. A great example of this is Turrison's trees, which were planted along a roadside by Turrison, I believe back in the 1930s. And what Turrison did was collect trees from all the way in northern Sweden, all the way to southern Sweden, and he planted them on the side of a road in order of their latitude. So, and every fall you can watch the northern trees lose their leaves earlier in the fall, and the southern trees lose their leaves later. And in the springtime, the southern trees put their leaves back on earlier than the ones later, or than the ones from the farther north. And this illustrates clearly adaptation along environmental gradients, and especially with latitude. So what I've been working on for the past over a decade now is switchgrass, Panicum virgatum, which a lot of you know about. It's a potential bioenergy crop and can be useful for both biofuels and bioproducts. One of the key great things about switchgrass is it's native to North America and it grows on marginal land, so it doesn't need a lot of extra fertilizer. And being a native North American grass means that the region in which it grows and where we do our experiments is in the native zone of the species, which allows us to really look at things like local adaptation and how switchgrass adapts across geographic space. And also there's this history of local adaptations in switchgrass dating back a half a century. And so we can look at those old experiments and glean from them ideas going forward of how to conduct experiments with switchgrass. And that's what I'll tell you about now. So if we think about the United States or North America in general, there are these large environmental gradients, water deficit going from east to west, and then temperature stress going from north to south as generalizations. And so if we want to study adaptation across those regions in a plant species, for example, like switchgrass, we would collect from plants all over that region and then studying them in common garden experiments. And that's exactly what Calvin McMillan did back in the 1950s and 60s. And Calvin McMillan was a faculty member originally at the University of Nebraska-Lincoln, where he planted a common garden with switchgrass collected all from across eastern North America. And he looked at the days in which the switchgrass flowered. And then then he got a job at the University of Texas at Austin, and he dug up all of the switchgrass plants from the common garden, took them down to Austin, Texas, and planted them there. And this is the data from Austin, Texas. And you can see across the state of Texas, there's actually a 15-day difference in terms of, or 15-week difference in terms of flowering time of the ones from the panhandle of Texas versus down on the Gulf Coast. And we think this is mostly due to local adaptation to those different regions of the country. So if we plot out Calvin McMillan's experiments and look at the latitude of origin versus the first day of flower of the plant, we can see that there's a strong correlation between these, where in Lincoln, Nebraska, you see that as you get to higher latitude, you have an earlier flowering time, and the same thing in Austin, Texas. And the relationship holds in both of those field sites. So based on this, the results of these classic experiments, we wanted to really understand the genetic basis of this difference across space. And so in order to do that, the first thing we did was start to target different ecotypes. And these ecotypes are, I'll use this terminology. This is the old terminology, which has been revised now with the release of the genome paper this year. But what we have is two major ecotypes, the upland ecotype and the lowland ecotype. And the upland ecotype typically is more drought tolerant, it requires more nitrogen, and it's more cold tolerant, and generally occurs in farther north in the United States, whereas the lowland ecotype is more flooding tolerant, requires less nitrogen, and is oftentimes more pathogen resistant, both to fungal pathogens and to virus pathogens. And so there's these two ecotypes that occur out there in North America, as well as there being this like latitudinal gradient of adaptation. And so we have to keep both of those in mind when we're studying this system. So just to illustrate those differences among ecotypes and populations from different areas, here are switchgrass plants growing in a common garden in Austin, Texas. This plant over here is a lowland plant from Austin, Texas area. And then this plant, an upland plant from Lincoln, Nebraska. And you can see that there's big effects of growing in different environments. This plant never gets as big as the southern lowland plants, but in Texas, it's very much dwarfed by the high temperatures and low soil water availability in that region. So in order to understand the genetic basis of adaptation across this large latitudinal gradient, back when I was a postdoc, I set up a genetic mapping population where I took 4 different lines to build a genetic mapping population for switchgrass. This includes Dakota from North Dakota, Summer from Lincoln, Nebraska, and then 2 lowland lines from Texas. So these are upland lines in blue and lowland lines in orange. And what I did was I crossed together the lowland line times the upland line. Sorry, the VS16 should be over here. And at the back here, it's Summer VS16. I apologize for that. And then this lowland line by this upland line, Summer VS16. And those crosses yielded F1 hybrids that were a hybrid between upland and lowlands. And then I intercrossed those F2 hybrids to make an outbred mapping population. And so this founded the basis of our first set of common garden experiments that we used to study the genetics of local adaptation in switchgrass. So we set up this common garden experiments for this mapping population all across the United States, or the central United States. And we planted these plants, clones of these plants, into 10 locations, all the way from southern Texas and Kingsville, up to South Dakota, and over to Hickory Corners in Michigan, where the Kellogg Biological Station is. And we monitored these plants over the last, I guess it's 6 years now, because we planted them in 2015. And we've collected a ton of data on them, including biomass data every year, and different sets of phenotype data every year as well. And from that, we were able to identify loci that contribute to adaptation to these different regions. So I'll just show you a quick example of that. So this is one of the many loci that we found, or QTL, that we identified in these experiments. And we could look at the actual allelic effects of these QTL across space. So what I have here is the 10 different field sites, and then each one of these bars is the allelic effect of this one locus at each of those field sites. So you can see that this locus has a big effect in the north. It makes the upland allele makes the plants bigger, and it has little or no effect down in the southern United States. And we think that this locus may be involved in overwinter survival or preventing damage of plants during the wintertime. And so it could be a locus that's involved in cold tolerance, which is very important in the northern United States. So we can actually plot out the effect of this locus across space, the allelic effects, and see that it has a large effect up in the northern United States and then little effect down in the southern United States. And so this is taking the data from our 10 field sites and then interpolating it across space, matching up with the climate data available across this region. So that's mostly what we've done so far with this mapping population, or what we published so far with this mapping population up through 2019. But then we came to the realization that it would be a great system for understanding the fungal pathogen resistance. And we thought that it would be a great system because we knew that there was differences in resistance between upland and lowland plants. And we knew that the lowland plants were typically more resistant to fungal pathogens. And we focused our efforts mostly on rust to start with, because we knew from a study back about 6 or 7 years ago that rust could reduce ethanol yields up to 55% in switchgrass plants. And the rusts are pretty wild because they show up every year. We think that they're killed off in the winter, and we think that they come up to the Midwest from the Gulf Coast refugia through wind-borne spores every summer. The rusts have a very complicated life cycle. They have 2 hosts and 5 spore types. There are 5 different species of rusts that infect switchgrass, but there's one that shows up pretty much everywhere which is this Puccinia novopeniceae. And this one is widespread, whereas the other ones are much more rarer. And we actually don't know what the alternative host is for this rust species. We think it might be some kind of euphorb, but nobody's actually identified the alternative host. But we know it shows up on switchgrass every year and can cause a lot of damage to switchgrass, and especially damage the plants before harvest in the fall. So here's some examples of different stages of rusts. So all of the work that I'm going to tell you about from this point forward has been led by a really stellar postdoc in the lab, Acer Van Wallendale. You can tweet at him here if you want to ask questions to him or send him an email. Acer does everything a professor does. He teaches classes. He runs aspects of the lab. Essentially, he has a lab within the lab going. And so he started doing this work back in my group in 2018, where he's focused on collecting data on rust infection at 8 of our field sites across the range. And from the data they collected together, from data he collected and from field technicians, he got rust scores from 149,000 plant by time interactions. So 8 field sites by 3 years over a 14-week time period by 471 genotypes. So we have a lot of data on rust infection across multiple years. And then he used that to conduct QTL mapping. And one of the really interesting results that Acer got out was that he had 2 large effect QTLs. So this is the summation of LOD scores from QTL mapping across the 4 northern sites versus the 4 southern sites. And in the northern sites, he has these 2 large effect QTL, but in the south, you don't really see that. And so there is this big genotype by environment interaction where things that lead to susceptibility in the north do not contribute to susceptibility or resistance in the south. And so we really needed to take this into account when we're thinking about pathogen infection. And so these 2 loci together contribute to about 34% of the difference in pathogen resistance of the variation in pathogen resistance. And they also contribute to about 44% of the difference in biomass. So there seems to be a locus that either directly contributes to differences in biomass by preventing rust infection, or is linked to another gene that contributes to biomass. And so we don't really know what's causing that, but this is a locus that you could imagine that breeders would want to select upon. One of the interesting things about these 2 loci is that they have epistatic interactions. And if you look here, here's the genotype for one of the loci versus the genotypes for the other loci. And in Missouri, we can see that when you have one of the upland alleles, this Dakota allele, and one of the lowland alleles, the AP13 allele, together, when they're combined together into a genetic background, so from one locus and the other locus, you get a lot more rust infection. So you get a much more susceptible plant. And that also impacts biomass as well. And then up in Michigan, we actually see the opposite pattern. So the plants actually become less susceptible to rust with that combination. So there's some kind of complex epistasis that also interacts with the environment to dictate susceptibility of rust in different locations across the United States. So we have a couple different ideas for why this might be. There either could be different pathogens in the north versus the south, like there could be more strains of rust in the southern US. We know that it's the same rust species that's the primarily dominating across regions, but we're currently doing population genetic analyses of that main rust species in order to understand if there's different strains. There are also, this pattern could also be caused just by genotype bioenvironment interactions, and there's precedent for this where there are rust resistance alleles that work well under some conditions and then often at higher temperatures break down in terms of their efficacy of resisting the rust pathogens. So the whole situation can be quite complex. Okay, so that was just a simple cross between 2 upland and 2 lowland plants. And so you could imagine that the actual genetic diversity that's out there in switchgrass might be quite different from what you could see in just that simple genetic cross. And so in order to understand whether there are other loci, like really common alleles that actually contribute to differences in rust susceptibility across space, we took advantage of this population diversity panel of switchgrass. And this diversity panel was collected over many years. And then the Joint Genome Institute and Hudson Alpha has worked to sequence or resequence all of these tetraploid and octoploid lines, which we've now planted into common gardens as well. And those common garden experiments are now located at many different locations, including the Great Lakes Bioenergy Center, which I'm part of, in Michigan, the Fermilab, a number of university field stations. The CBI has 2 field sites as well in Georgia and Tennessee. And then there's a couple USDA field stations in Texas. And so it's a really big collaborative effort where we have this diversity panel planted in all these different locations. And it's available now if you want to go and measure interesting phenotype or look at different microbes on this diversity panel. As Jeremy talked about a little bit early in the introduction to this session, this was recently published in Nature. All of the work was done and clearly separated out these different groups of switchgrass into different genetic groups. And we could see that the ones from the northern US, especially these Midwestern ones, they survive winter much better than ones from the southern United States, especially the ones from along the Gulf Coast. And so we're seeing that there's big abiotic differences in terms of between these different types of plants, the ones from the north and the ones from the south. But there's also probably big biotic differences. And so that's what I'm going to tell you about now. So we've surveyed rust intensity now for a number of years across these field sites, and we're in our 3rd year of surveying now. And as you can see at the field sites from down here in Kingsville, Texas in the south to Brookings, North Dakota in the north, we do see like a wide variation of rust infection. So scored from 0 where there's no infection to 10 where there's a high level infection. We see a lot of infection in Columbia, Missouri. And one big caveat of this is that this was all scored by different field technicians. So it could be that some of the field technicians like score things a little bit more infected than other technicians. But when we do have variation for this, that allows us to conduct genome-wide association studies within field sites to identify loci that control susceptibility versus resistance in those areas. And so with that, we've conducted genome-wide association studies across these different field sites, and we've actually identified major loci that control rust resistance. We see different loci showing up at different field sites, so Brookings, South Dakota versus Lincoln, Nebraska. But actually, in this case, we actually have the same type of gene, this Acyl-CoA synthase 8 type gene showing up at both of those field sites as being highly associated with rust resistance. And so I could show you plots of all of these different field sites, but needless to say, it's complex. There's different loci, some of which consistently show up across a lot of field sites as being important, and some which blink in and out depending on what field site you're located at. In addition, we noticed that there was a lot of smuts that had come into our plants later on. Smut usually settles in about year 3 or 4 for switchgrass, especially in the north. And so we conducted genome-wide association studies of that. And at most field sites, we didn't come up with an association. But at the GLBRC field site up in Michigan, we actually did have a strong association with a locus on this chromosome 2. And so we'll be exploring that further to see if there's anything going on this year, because we'll still be collecting data on SMUT going forward. So hopefully we'll have a nice paper on both SMUT and Rust genome-wide association studies coming out sometime in early next year. So a lot of you are very interested in microbiome, not because of the pathogens, but because the microbiome can actually perform positive functions for its host. And so as many of you know, the microbiome, as we saw in a previous talk, can actually keep pathogens at bay, can help to help in nutrition, all kinds of different things. And so it's really important to actually understand the whole microbiome. And Acer got interested in looking at the fungal microbiome in general and understanding how it changes across the season, and whether we could glean any insight from that into understanding differences between different switchgrass genotypes and different field sites. And so Acer recognized that the host can impact microbiome colonization and growth. And so we imagined that we could have different microbial communities across different genotypes, but we just didn't know, because nobody had really tested this in switchgrass, especially for the above ground phylosphere community.
David, just about 5-minute warning, okay?
Okay, thanks. And so we conducted this experiment where we had microbiome succession over a field season, where Acer collected from 100 different genotypes from this diversity panel at 5 different time points, up at our GLBRC field site in Michigan, but then also at a couple other field sites in the southern and central United States. And we were interested in looking at the microbiome communities change over time, but then also how this will impact downstream bioenergy traits like deconstruction and conversion. And that work is still ongoing, but I can tell you about our results from the microbiome work so far. So with this work, we collected samples from the field and conducted ITS sequencing. Acer detected approximately 6,000 different species of fungi in the leaf phylosphere community, and from that started conducting analyses. And so just to summarize those results, he found that there was really strong differences in terms of the fungal community across time points. So early on in the season, the community was much different from later on in the season, and also saw that there was differences among these different subpopulations of switchgrass. So the genotypes, whether it's Gulf, Midwest, or Atlantic, also affected the what the fungal community was. And so using trajectory analysis, Acer tracked the fungal community across these different genotypes across time, and saw that there was, a lot of the genotypes were similar, but this Midwest population group had a quite different fungal community, and that changed with time in a somewhat different way than the other genotypes that were out there. In addition, there was big difference in the fungal community across field sites. Northern field sites were quite different from southern field sites in terms of what fungi were there. So finally, we've been trying to combine these results that's looking at the microbial community with drone-based phenotyping. And so along with collecting data on the microbial community in terms of the total microbial community, and then rust pathogens, smut pathogens, and viruses in collaboration with Carolyn Malmstrom at Michigan State University, we've been collecting drone-based hyperspectral data throughout the field season as well, that pairs up with that, as well as leaf level physiology. And so with all of that data together, we're hoping to build predictive models of microbial communities over time, and to look at whether we can predict fungal infection or rust infection before we actually see it manifest on the leaves. In addition, we did a survey this last summer where we collected from all of the tetraploid genotypes tissue for gene expression analysis, which we'll be using for expression QTL mapping. But one of the exciting things about this is, because this is RNA that we collected, we can also look at RNA viruses and look at whether different genotypes of switchgrass have different loads of RNA viruses, and whether we can even identify loci that control whether or not those viruses are present. So all in all, it's a pretty exciting time to be involved in this research. We have a lot of data that's come in that we've gotten results for, and there'll be a lot more data coming soon. And so look forward to seeing a lot more papers coming in the future talking about all of these results. Okay, finally, in conclusion, just to wrap it up, some of the main conclusions is that in our mapping population, there was epistatically interacting loci, and genotype bioenvironment interactions with those loci contributes to pathogen susceptibility across space. While we have identified major loci for rust, and we have now identified major loci for rust and smut resistance using genome-wide association studies, and we're curious to see whether we can identify specific genes that are involved in rust resistance from those studies. And we've started to track the fungal microbiome community over the year and see that we have effects of field site, genotype, and time of year all in that microbial community. And so I think it is an exciting time to be doing this research. And just to wrap up, big thanks to everybody that's been involved in all this research, especially the folks at JGI and Hudson Alpha for a decade now, especially Jeremy and John Lovell, who've done a lot of work with us for such a long period of time. It's been great. Funding came from Department of Energy, from individual grant on our rust work, as well as the GLBRC. Big shout out to Acer Van Wallendale, who's done incredible work bringing all these studies together, bringing in teams like Team Microbiome from the GLBRC to get involved so we could really start to study fungal communities in detail. And with that, I'll take questions.
Thanks, David. Appreciate it. The great overview and telling us about what's going on in the switchgrass fields. So if you want to stop sharing your slides, you can do that, or you can leave them up. It's up to you. Oh, thank you. So we've got a few questions here. Some detailed question here for you to start with. For the rust resistance loci you've identified and the smut ones, do you have data on how long these polymorphisms have been maintained in switchgrass, meaning in the population at large?
Yeah, that's a really great question, and I don't offhand know how long those polymorphisms have been maintained, and I don't know exactly how big that region is for our GWAS peak there, but that's a great thing that we should look at for sure.
Okay. More work needed on that. Have you tried doing any reciprocal experiments where you inoculate soil microbiomes from plants from the warm regions versus the cold regions, or vice versa? To see if there's any kind of direct beneficial changes to plants when in different temperature conditions.
Yeah, no, we haven't done any of that kind of inoculation work, especially across field sites. There's been inoculation work with soil, especially working on soil microbiome at the GLBRC with Greg Bonito's lab in particular, but we haven't done much work with phylosphere community and try to manipulate that in any way. We've mostly just been monitoring at this point and seeing this patterns of succession across time.
Right. Yeah. So those would be kind of interesting experiments if you could break them down into real sort of very specific experiments of combinations of microbes or enrichments from the different growing areas, natural growing areas in switchgrass.
Yeah, and I would say one of the interesting results, which I didn't talk about here, is that Acer actually identified a microparasite fungus, which often attacks rusts and was associated with lower amounts of rust in the leaves. So finding things like that, where maybe we can manipulate that microparasite and increase its abundance and thus decrease rust infection, could be something that could be highly beneficial. Although I just don't know how hard it would be to manipulate.
Exactly. The manipulation would be important there. So, and then another sort of genetics question here on the heritability. So what are you seeing in the heritability? Do you see differences between the southern and northern gardens in heritability for the rust or smut resistance?
I'm not so sure about the heritability itself, but we do map more loci, it seems like, in the northern US to be associated with both smut and rust resistance, especially with the rust. But I need to think more about that.
Yeah, that would be sort of a little bit counterintuitive, right? Because we kind of think about although maybe some mapping bias involved there too, right? Because if you've got a lot more active resistance elements going on in the south, it might be more difficult to specifically get QTLs out of those than what you're seeing in the north, where you have a sort of maybe a less biased selection towards those resistance elements. Definitely, seasonality is really important here and there. Okay, well, David, thank you so much. I really appreciate you coming today and telling us about what's going on in Switchgrass and both the bad and the good from the smaller eukaryotes here. And appreciate it. And so we're going to end now, folks, this session, and the posters are open, and also the lounges are open. So please drop in and say hello and see what's going on around the rest of the JGI and around the rest of the JGI users. So I appreciate it. Thanks again.
Thanks so much, Jeremy.